All right. Hi everyone, and welcome back to another day of Bio 148. It seems like when I was out here the other day, maybe the locusts were louder, or the cicadas were louder than what I was. So I'm going to talk a little bit about mutation again, and then we're going to talk about some evolution misconceptions, and then we'll talk a little bit about speciation, how it can happen. I'm not going to do the learning objectives for speciation today. I'll do them for tomorrow. Since I'm just going to introduce the topics today, we're going to add more on top of it tomorrow, and then I'll do the learning objectives tomorrow. Okay? All right, so let's go ahead and get started talking about mutations. Okay, so here on day 12, this very beginning part was a little bit hard for some people to hear. I don't know what happened. I think when it uploaded, like the cicada noise drowned me out. So what we had here was we had two different, we had a single molecule, and I think, and when I had that single molecule, I think these ones together, right? So this was like my original strand, right? And this was my original strand as well. And then I had made copies of them. So let's just take this, oops, and make it a new color. And then I think this one was also a new one, right? Okay. And here I had made a mistake, right? And I had talked about a little bit about how this mistake that I had made, right, could be fixed in two different ways. So I had originally made this A mistake, right? When I was trying to copy the sequence, I had matched an A with an A. And when another RNA polymerase came through to try to check everything, it solidified that mistake by changing this bottom one to a T instead of changing the top one back to a T like it should have. Okay, so we had a mutation on our new strand, and then another DNA polymerase that comes through to do fixing had fixed it so that it was a mutation and was present in both strands. Okay, so the molecule lined up in the complementary way, the way that it was supposed to, but this mutation got fixed. Okay, and in my original population, I had said that I had all A's, and then this new mutation, I called it a capital A, right? So remember that mutations can be good or bad. So think back to like the stickleback fish, right? So the fish having a mutation in the mutation in PIDX1, switch for the pelvis could be good or bad depending on the environment. We could also have that RNA polymerase mutation, right, in bacteria. And then we also have had the example of like the beans that we've done a few times by now. Okay, remember that mutations are random, that they can happen in the regulatory regions like the fish, in the promoters, or in the coding regions like in the RNA polymerase. Okay, these can happen in somatic cells, but when we're in somatic cells, that is not going to lead to evolution. Okay, something that happens in the somatic cells is going to be in me, but not in anything else. If a mutation happens in a germline cell that leads to the production of egg or sperm, that's when it can be inherited in the next generation. So if I am out in the sun too long and I get a mutation, even before I have kids, if I get a mutation that causes skin cancer, my kids are not going to get that because it's just going to be in my skin, okay? But if I have a mutation in the germline that is going to produce an egg, that's when I'm going to have that mutation that can be inherited. Remember here that evolution is a change in allele frequencies in a population over time, okay? And so mutation alone causes this change. So here in this population, I had 100% A to start with, and then after this mutation happened, I had changed my allele frequencies so that I had 8% of the capital A and 92% of the little a, right? So mutation alone can cause a change in allele frequencies. And then I think the video is fine for the rest of this. So just let me know if you guys ever see something that doesn't go, convert well or whatever. So I try to look quick, but I don't always have time to listen to the whole thing. And I checked that one in the middle of the video instead of at the beginning. Okay, so next I want to talk about, I'm going to open a new note for day 14. Okay, so today we're going to be on day 14. And we're going to talk about a few different things. So we're going to talk about evolution, misconceptions. A lot of these we've actually already covered, but I'm just kind of putting them all together. Okay, evolution, misconceptions, and how speciation happens. And speciation is when one species becomes two separate species. And then we're going to talk about how we define different species that currently exist on Friday. Okay, so let's start with evolution misconceptions. Since we've already covered a lot of these, I'm just going to pull up the learning objectives to start with. Okay, so here we go. Here are my misconceptions about evolution. Okay, so here I've listed out kind of 6 different statements about evolution, but then right below them you'll see is the misconception that kind of belongs with them. Okay, so note that evolution is changes in the genetic makeup of populations. Okay, so a lot of people think that evolution is causes changes in individuals, and that's just not the case, right? So evolution, remember, is the change in allele frequencies in a population over time, right? So natural selection is going to act on individuals, right? Drift might remove some individuals and things like that, but evolution really is at the population level, okay? The second misconception is that adaptations occur in response of a need. I need 2 sets of hands. That would be amazing. If I had 4 hands, I could do so many more things. But that isn't, just because I have that need doesn't mean that that's what I'm going to get, okay? Mutations are going to always be random, okay? They do not pop up because there's a need in an organism, right? We talked about this a little bit when we talked about rifampin as well, is that those mutations could pop up whether there's rifampin around or not, but the natural selection is only going to act on those populations and keep them in the population if rifampin was around, right? The next one is that traits can be lost over time. So evolution does not always result in more complex organisms. My favorite example here is cavefish have lost their eyes. So you guys can Google this if you want. And there's lots of examples of this. But, yeah, okay. So one thing that has happened is that cavefish have lost their eyes. It's better actually for them not to have it, and they have kind of a covering over where their eye should be. So there's kind of an eye socket, but no actual eye, because they live in the dark. They don't actually need to be able to see, and it's just a place for infections and things to happen that isn't necessary, right? So evolution does not always result in more complex organisms. Traits can even be lost, okay? For number 4, small changes in switches especially can have really large phenotypic effects. So think about like something like PITX1, right? Think about our PITX1 gene, where a single mutation, right? Here's that PITX1 transcribed region, right? We have some switches here, 2, 3 switches. Remember that we could have a single mutation right here, right? And a single mutation could make it so that there's no jaw, no pelvis, and no pituitary gland, right? And if we don't have, especially this one, right, then we can't have anything else forming, right? So a single change, just one single change, can have a huge effect. One single change can have big effects, right? So some people think that we need really big changes, lots and lots of changes at the genetic level to have a large phenotypic change, but that's actually not true, especially when a single gene might have a role in lots of different things, okay? Next is that natural selection is not random. So remember that some individuals survive and reproduce more due to the traits that they have in their DNA. So some people think that natural selection is a random process, and it's not, right? So natural selection is not random. Evolution can be random. Mutations definitely are random. Drift is random, but natural selection is not random, okay? And then the last one is some people think that natural selection results in perfect organisms, but remember that this isn't true for lots of reasons. So one of the things is that we have to remember that there's going to be mutations, right? There's going to be genetic drift, and there's going to be gene flow that might not have anything to do with things that might be perfect organisms. And natural selection itself has limitations as well. So here it's showing some examples of natural selection being limitations. So again, it would be great if I had 2 sets of arms. I don't have that. I'm not a perfect organism, right? None of us are perfect, and neither are any of the organisms around us, okay? There's lots of constraints depending on like which genes we have, right, is a constraint. There also are trade-offs. So we have bipedalism in humans, so we can stand upright, but that causes a lot of issues for giving birth, as well as even babies' development. So human babies are born a lot earlier in their development than a lot of other mammals. So just think about how long you have to kind of carry a baby around versus if you've ever seen a horse being born or kittens or anything else, and how quickly they can move compared to how quickly humans can move, right? And that's largely because of challenges introduced by the ability to walk on 2 legs instead of on 4, okay? So those are just some misconceptions about evolution. And I thought that that was kind of a shorter lesson, so I wanted to talk a little bit today also about how speciation happens, okay? So we're going to just do a little line. Actually, I'm going to go ahead and, yeah, let's just do it. I don't think we're that far. We're just going to do a line. So the question is, how does one species become 2 separate species? And this is a question that has actually interested me personally for a really long time. It's what I did my PhD on and some of my postdoc work on as well, okay? So there's 2 kind of overarching categories, okay? There's allopatric speciation and there's sympatric speciation, okay? So there's kind of 2 different types. Allo, so think about allo, if you break down that root, it's going to be different, okay? So allopatric speciation will happen in different geographic locations, okay? Sympatric, think of sim, is going to happen in the same geographic location. Okay, so when we're talking about allopatric speciation, we kind of have 2 types of allopatric speciation. One of those types is by vicariance, okay? And vicariance is when you have a single population, okay? So let's just we're going to be watching some frogs for a friend here in a little bit. This is a frog. I'm going to do a little face for it here in a minute. Okay, and something happens, maybe the water recedes in a pond and there was a little bit of a sand area, and many years of drought, maybe there's a big change in the weather pattern or whatever, has divided one population into 2, okay? So vicariance is when we have one population divides into 2 populations. And then those 2 populations accumulate differences. right? And these are going to be genetic differences. So it could be that drift acts separately on these two different populations, and a lot of neutral mutations happen in both populations and eventually lead to some isolation, and we'll talk about that a little bit more tomorrow, okay? It could be that that there's natural selection and that there's different forces on these two different sides, right? So maybe there are two different types of predators on the two different parts of the pond, and so they're under different pressures, okay? So just think about that, how we could have both natural selection that is kind of a non-random process acting on these populations, or we could even have non-random like natural selection, or random processes like genetic drift and mutation just starting to accumulate and result in splitting these populations, okay? So vicariance is one, and the other one is dispersal. Okay, so in dispersal, again, you have that same initial population. Actually, maybe I'll go back. Okay. So you have that initial population, and a few of these individuals, okay, go and inhabit a new area where nobody else is already there yet, okay? So a few individuals go to this new area. Okay, so they go to a new area. So note that this is different than gene flow because these are the first individuals on that population. So this dispersal, okay, is where some individuals go to a new location. Okay, that doesn't already have any there. And then again, then those populations accumulate genetic differences. And this is the same up here too, okay? Is that we have to have those genetic differences. So let's move that down just a little bit, right? Just to specify that these are going to be genetic differences. So some of them we can easily see and some of them maybe we can't, okay? So here we have allopatric speciation. Note that this happens in two different locations. So we have these populations in the two different areas in vicariance, and then we also have them in two different areas with dispersal, okay? And this is different than sympatric speciation. So note that with allopatric speciation, we have different geographic locations happens first, and then we accumulate differences, okay? In sympatric speciation, we're going to happen in the same location, okay? So the first thing that's going to happen is that we're going to have genetic differences. Okay, and so again, we have kind of this in two different flavors. Okay, so one you already know about actually is disruptive selection, actually. Okay, so remember with disruptive selection, we can even think back to those beans, right? Where maybe it's good to be white or dark, right? But in the middle is bad, right? Okay, so there's pressure not to be in an intermediate like level of white and dark with those beans, right? So it was good to be white, it was good to be like really dark brown or black, right? And really bad to be in the middle, right? And so these differences are because of genetic differences, right? We talked about that before too, is that these were due to genetic differences. And so genetic differences happen first to make it so that you are either white or dark, okay? So that's one of them. So these differences are due to, oops, let's do it this way. Sorry, that was really messy. Okay, so genetic differences yield character differences selection is acting upon. And this can be lots of things. This can be size. So you guys just did that that case study with the sawflies. So it seems like sawflies have a size difference that really makes a big effect, where small ones are mating with small ones and large ones are mating with large ones, and there's some differences in size that are causing disruptive selection in Lecontii and Pinetum, right? So you learned that with both the saw, but it's also important to note that that actually is happening with their mating as well, and that's kind of up and coming research is going on in that lab right now. Okay, so we've got disruptive selection, but then we also have is one type, and then the other type is ploidy changes. And I think I just looked around to see if I could spot a strawberry, and I just did. So I'm going to show you guys an example of this, okay? So let's go back to the camera. And right here, you see that red little thing? So this red little thing here is a strawberry, and it's a wild strawberry. I don't remember. I think this one is actually the European variety. Not our native strawberry, but you can see the leaf shape and everything looks a lot like a strawberry, and so does this with the seeds on the outside. Okay, I just learned a fun fact is that the each little strawberry seed, since it's on the outside, each one of those is considered a nut, apparently. I don't know, something to look up. It's kind of fun. And then I was going to see if I could find, sorry, the garden is a mess because I've been spending all my time working on stuff for you guys. Okay, let's see if I can find a strawberry. I swear I saw one in here the other day. I bet one of the girls ate it. Okay. Okay, these ones just got transplanted, so they're a little bit sad. But here's a strawberry just coming on. And you can see that this strawberry that is still green, and it's even this strawberry right here is bigger than that strawberry that you saw on the other side that was already turned red, right? So that one that was turned red was like this size of the small one that just started, and the strawberries are going to keep on growing and growing and getting bigger and bigger. I don't have any ready and ripe right now, okay? But the difference between the strawberry that I showed you first, that's a wild strawberry, and these ones here is actually a change in ploidy. So these strawberries and those strawberries cannot mate with each other, okay? And it's because of a change in ploidy. So what ploidy means is like how many copies of each chromosome you have. Okay, so I think I've thrown out this word diploid before. So the wild strawberries are diploid, okay? So wild strawberries, just gonna are diploid. So they have 2 N, okay? And then these other strawberries that are like, that have had artificial selection done on them, okay? So the ones we eat and the ones in grocery stores, you can eat some native strawberries too. Some of the wild strawberries. This one is not very good. I think it's the European one. Okay, the strawberries we get at the market are actually 8N. Okay, so for every chromosome that they have, like they have a gene that's for like the red gene. These ones have 2 copies of all genes. These ones have 8. Okay, they're octoploid, right? They have 8 copies. And this difference makes it so that these guys cannot make babies, right? They can't mate with each other, okay? And they're 2 different species. And this happens pretty often in plants, especially plants that we have been selecting different traits for, is a lot of times there's been changes in ploidy, okay? And we'll come back to that a little bit more actually in our last unit too, when we're talking about mitosis, and we'll make a connection to this. So remember that we're going to come back to this. And if things don't happen correctly during meiosis, so meiosis is when you're going to be making the gametes for the next generation. When things don't happen well in meiosis, then you can get this change in ploidy, okay? And plants do this pretty often, actually. Okay, so I just wanted to introduce those two because I thought that tomorrow having all of the speciation things in one day was a lot, so I just wanted to break it up. And we will talk about the learning objectives for speciation tomorrow. Okay, bye guys.